Hello everybody and welcome back to Unit 1 Review. Today we're going to be talking about topic 1.5, which concerns the origin of cells. So before we get started, I just really quickly wanted to outline the essential idea that IB presents throughout 1.5. This essential idea is that there's an unbroken chain of life from the first cells on Earth to all cells in living organisms today. So outlined on this slide are just the things that we need to know for 1.5 when it comes to the test. So make sure to take it upon yourselves to kind of dig deeper into understanding all the different things outlined on this slide. So getting into cells and how cells have developed, we have two main theories. The first theory is the theory of abiogenesis, and the second theory is that of biogenesis. So biogenesis essentially means making new living things. More specifically, this is the theory that living things only come from other living things through reproduction. Abiogenesis, which is sometimes referred to as spontaneous generation, means life coming from non-living things. So these are the two main theories that revolve around cell development and the way that cells today within human bodies and within different organisms have come to develop over time. So, evidence from Louis Pasteur's experiment proved whether or not abiogenesis and biogenesis were accurate theories in kind of figuring out cell development and how cells have developed over time. So, evidence from his experiments essentially proved that spontaneous generations, otherwise known as abiogenesis, does not now occur on Earth. What that means is that the formation of living things from non-living things isn't something that happens in Earth or on Earth today. So the most famous experiment that kind of proved this was when Pasteur placed a variety of different flasks, as you can see on the screen right there with that kind of curved neck, and he boiled the broth inside of the flasks, and he left some of them unboiled just so that he could have a control group. What he noticed immediately was that the broth that had been boiled kind of retained its original stance, or it retained its original form as a broth, whereas the one that had been unboiled kind of began to develop different deformities and different organisms within it. So what he proved from this, or what he kind of figured out from this, was that the air kind of may have infected the broth into inducing living organisms within the broth. So from there, Pasteur decided to take it a step further to further analyze and develop evidence for either abiogenesis or biogenesis. And so what he did is he broke the flask top, that curved flask top that you can see there on the screen, from a couple of the flasks, or a set of the flask that he identified as experiment number 2, and he kept some of the flask tops on a group identified as experiment number 1. So after observing these two flasks in these two experiments, he noticed that the flask that had the curved neck remained the same, and they didn't actually grow any bacteria or any fungi, whereas the flasks with the snapped necks began to have organisms apparent inside of the broth, and the broth began to decompose. Essentially, what this did is it led to Pasteur publishing his results in 1860 and subsequently repeating them with other liquids, including urine and milk. And so what his intent was in doing this was further proving whether this was applicable to all living things or to all things, or whether it was just that one situation with the broth. So he furthered his analysis by testing these different liquids. So in doing this, he disproved the theory of abiogenesis, as he concluded that the swan necks prevented organisms from the air from getting into the broth or other liquids, and essentially this proved that no organisms appeared spontaneously. So now we're getting more into non-living synthesis, which is essentially how cells have developed over time prior to human life and prior to organism life. And so there are 4 distinct stages in the development of life from non-living material. So the first one is the production of simple organic molecules from inorganic molecules. And this idea kind of came to be because of the Miller-Urey experiment. This proved that it was possible, essentially. Um, what Miller and Urey did is they passed steam through a mixture of methane, hydrogen, and ammonia. The mixture was supposed to be representative of the atmosphere in early Earth development. Then the scientists released electrical discharges, which were used to stimulate the lightning that was occurring on Earth when it was when it first came to be a planet. So when they did this, they found that amino acids and other carbon compounds needed for life were produced. So from that interaction, from that kind of gas mixture and electrical mixture, there were things that were formed, and essentially those are things that are now integrated into cell life and to the way that cells function. So this was an example of how a non-living thing created a kind of living thing, or something essential for life. So the second distinct stage of development from life to, or from non-living things to life, is the assembly of carbon compounds into polymers. So this kind of idea is representative. We can see it through deep sea vents. What deep sea vents are cracks in the Earth's surface, which are characterized by gushing hot water, which carry reduced inorganic compounds and chemicals such as iron sulfide. So what these chemicals represent is the readily accessible supplies of energy, a source of energy for the assembly of these carbon compounds into polymers. So these deep sea vents give us a little bit of insight into this second distinct stage in the development of life from non-living materials. So for the third distinct stage in the development of life from non-living materials is the formation of membranes. So this essentially came to be because of phospholipids or other amphiphatic, sorry about that, carbon compounds, as they were the first carbon compounds and would have naturally assembled into bilayers. So different experiments in the past have shown that these bilayers readily form vesicles which resemble the plasma membrane of a small cell, as you can see to the far right of the screen. So essentially, this would have allowed for different internal chemistry from that of the surroundings to develop, which allowed for isolated internal chemistry. So what this what this did is it allowed for things to begin developing that differed from their actual environment, which kind of led to that production of the cell and the way that the cell works. The fourth and the final distinct stage in the development of life from non-living materials, is that polymers could self-replicate, or otherwise known as inheritance. So what this is, or what this concerns, is the idea that living organisms currently have genetics made of DNA, and they use enzymes as catalysts in order to replicate DNA and to be able to pass genes off to offspring. Enzymes are necessary for this process. However, for enzymes to be made, genes are needed. So the solution for this may have been in an earlier phase of evolution when RNA was a genetic material. RNA was able to store information in the same way as DNA, but it is both self-replicating and can itself act as a catalyst. So what this essentially means is that the production of genetic material kind of led to the development of reproduction and inheritance between cells, which kind of gave structure to the way that cells are now and to the way that they reproduce and that they restructure themselves now. So going along with this, we kind of get into the universal genetic code and the different aspects that kind of make all forms of DNA and all cells the same. So all organisms in the world kind of coincide or kind of have this gene code as their genetic code. Essentially what this does is it allows for DNA within organisms to be susceptible to that kind of development and that kind of evolution that we've seen over the millions of years that organisms have been kind of growing and changing into what we know them to be today. So the endosymbiotic theory kind of helps us to explain the evolution of eukaryotic cells, which is what I'm going to cover on this slide. So, what this theory states is that the mitochondria were once free living prokaryotic organisms that developed the process of aerobic cell respiration. So, what that meant is that larger prokaryotic cells that could only respire anaerobically took them in by endocytosis. So, instead of killing and digesting the smaller prokaryotes, they allowed them to continue living in their cytoplasm. This was essentially a mutualistic relationship between that smaller prokaryotic cell, which is now identified as the mitochondria, and the larger cell, which is now identified as the whole cell. But essentially, as long as the smaller prokaryotes grew and divided as fast as the larger ones, they could persist indefinitely inside of those larger cells. So, according to the theory of endosymbiosis, they have persisted over hundreds of millions of years of evolution to become the mitochondria inside of eukaryotic cells today. So, the larger prokaryotic cells and the smaller aerobically respiring ones were in a symbiotic relationship in which both of them benefited. Like I said before, they were in a mutualistic relationship. The cell would have been supplied with food by the larger one, and the smaller cell would have carried out aerobic respiration to supply energy efficiently to that larger cell. Because of these circumstances and because of this mutualistic relationship, natural selection favored the cells that had developed this endosymbiotic relation. So, the endosymbiotic theory also explains the origin of chloroplasts. So, if a prokaryote cell had developed photosynthesis, was taken in by a larger cell and was allowed to survive, grow, and divide, it could have developed into the chloroplast of photosynthetic eukaryotes. So what this means is that those smaller prokaryotic cells, which are kind of in the same realm or in the same kind of area as the development of the mitochondria, however, those cells photosynthesized, those cells were able to be integrated into the cell, the larger cell, the same way that the mitochondria was, which ultimately resulted in that development of the chloroplast within photosynthetic eukaryotic cells. So again, both of the organisms in the endosymbiotic relationship would have benefited from the chloroplast in the larger cell. So essentially, the endosymbiotic theory kind of epitomizes the development of chloroplast, eukaryotic cells, and the mitochondria, and it kind of just explains this in relation to cell development, and it's a theory as to why cells have developed the way they have or how they've developed the way they have. So this is just one theory as to how cells have come to be and how we've come to kind of give certain attributes to certain cells. So evidence for the endosymbiotic theory kind of revolves around these major concepts outlined by the mnemonic device Mad Doctor. So the first one is membranes, which essentially concerns the double membrane bound organelles. The second one is antibiotics, which concerns susceptibility. The third one is division, which concerns the mode of replication within these cells. The fourth one is DNA, which talks about the presence and structural composition of these cells. And then the last one is ribosomes, which concern the size of the ribosomes and the way that they've developed the cell. So all these components of evidence contribute to how we know that the endosymbiotic theory is something that is feasible and is something that is realistic in kind of understanding what specifically has happened to cells and how cells have specifically adapted over time. Okay, so we finished with all the content, so good job with that. And now we're just going to get into a couple of practice questions. So the first practice question I have for you is, what is evidence for the endosymbiotic theory? So the endosymbiotic theory is what we just went over. Oops, sorry about that. It's what we just went over, and what I want you to do when you look at this question is I want you to immediately look at the responses. So in the responses we can see RNA, we see meteorites, we see amino acids, and we see the mitochondria. So just based off of these first components and looking back at the evidence that we outlined here on this slide with the mnemonic device Mad Doctor, we can kind of determine which one is already the answer, just based off of that mnemonic device and looking at what the endosymbiotic theory was. So really quickly, I'm going to go over again what the endosymbiotic theory was. So the endosymbiotic theory concerned the development of the mitochondria and the chloroplast and the development of the eukaryotic cell. So all of these answers kind of talk about the formation of cells, but they don't necessarily talk specifically about the endosymbiotic theory and that kind of mutualistic relationship that the prokaryotic cells had with one another, which led them to develop the way they have today. So the only answer that would be right is D, which talks about the mitochondria, which was outlined in the mnemonic device Mad Doctor. So the second question I have for you guys is describe the origin of the eukaryotic cells according to the endosymbiotic theory. Again, we have the endosymbiotic theory, and as I just explained, the endosymbiotic theory concerns the development of cells in terms of the mitochondria and in terms of the chloroplast, and overall the development of eukaryotic cells as outlined by the question. So for this question, what I'd want you guys to do is I'd want you to look at the mnemonic device that I outlined for you. And I'm really sorry about my writing. I'm not the best. But once you have this written out, you can kind of see what you would be able to talk about. So for the first one, it talks about the membranes. And I'm just going to put the first couple of letters and move on to explaining. So with this, what you could talk about is the membranes. You can talk about how organelles became double membrane bound. You can talk about specifically the mitochondria and how that kind of contributed to the development of eukaryotic cells. And if we look at this question, I forgot to mention earlier, it's 4 marks. And there are 5 outline components within this mnemonic device. So if you choose 4 of those that you know best and that you feel most comfortable talking about, you could essentially hit all 4 of those marks without losing any points or getting anything wrong. So the first one that you could do is about the membranes and talking about the mitochondria. The second one is talking about the antibiotics. Here is where you could kind of just discuss susceptibility and the way that antibiotics have come to show how eukaryotic cells work. If you're comfortable speaking about that one, you can go ahead and speak about that one. For the third one, it talks about division as a mode of replication. So for this one, what you could talk about is you could talk about how cells have come to kind of develop and how they kind of work with one another or work individually to reproduce. So in this point, what you would talk about is you talk about how cells come from other living cells. And you can kind of explain how natural selection contributed to the development of eukaryotic cells because natural selection favored these cells, which led to the increase of the survival of these cells, which is a part of division. So the second and last one is DNA. With this one, you can kind of just talk about how DNA has contributed to the development of eukaryotic cells. So what you could talk about is how that genetic information has led to these cells being able to function properly and these cells being able to code for more complex things and to carry genetic information in order to successfully produce organisms and things that contribute to the society we know today. And for the last one, what you could talk about is ribosomes. So with this one, what you could talk about is you could talk about how ribosomes contribute to the process within eukaryotic cells and how this has kind of led to the development of eukaryotic cells and organisms in general, living organisms in general. And that would go ahead and give you that 1 mark. So I'd like to remind you guys for free response questions, make sure to be as brief as possible. So what I would suggest for this question specifically is kind of doing 4 sentences, 1 per each point that you want to talk about for those 4 marks. So you could do like 1 for the membrane, 1 for division, 1 for ribosomes, and 1 for DNA. Or you can kind of work to combine those. So just making it as brief as possible and as kind of straightforward as possible will give you the best chance of being successful when your score is looking at your test and looking at your answer. So just keep that in mind, especially when you're just looking at paper two in general and looking at the questions within paper two in general, just because you want to just get the answer out there. You don't want to add a lot of fluff, just so that the answer can be straight up. So that's all I have for you guys today for topic 1.5. Hopefully you guys learned a little bit more about the origin of cells and how cells have come to develop over time. And hopefully these practice questions have given you somewhat of an idea of how 1.5 is going to be applicable in regard to the test. So we'll see you guys later, and hopefully you learned something.